Welcome back. In this video, I will discuss about microscope and microscopy. The word microscope is derived from the Latin word micro, which means small, and the Greek word scopos means to look at. Microscopes are instruments that are used to magnify small objects. Microscope is an optical instrument consisting of lens or combination of lenses for making enlarged or magnified images of minute objects. And the microscopy is the science that studies microscopic techniques. If you are a microbiologist, then definitely you have seen a microscope in your university laboratory. Yes, you are right. Just because of this instrument, you are in this course. Without this instrument, the progress of this branch of biology would be much slower. The fact is that although humans exploited the power of small living creatures before the discovery of microscope for wine and bread making, but who actually played a key role behind this sugar to alcohol transformation was unknown. For the first time, Antony van Leeuwenhoek observed those tiny creatures using his own handmade microscope. And after that, gradually this tiny world get unfolded. So the contribution of the microscope for the development of this branch of biology is obvious. So we have to study the microscope first, because we are going to study some tiny creatures that are invisible, and they are called microorganisms or simply microbes. But before we start, we should know how it works, how many types of microscopes are there, and how to use them. In order to understand microscopy and microbes, first you have to learn some basic physics of light and interactions of light with the medium. Do you know why? Because in a dark room we can't see anything. I hope now you understand the importance of light in microbiology. So let's start. First, what light is. Light is a wave in nature with energy which have both electronic and magnetic properties. Both electronic and magnetic waves moving in the same direction but in the right angle. When it passes through the medium, several things can be happen, and because of that interactions, we can visualize some objects and some are not. I know you are also a biologist like me, so I will definitely not going to derive any difficult equations of light, but I will try to simplify those sections only which are really important. The type of interactions are as follows. The first one is called reflection. If the light strike an object and bounce back, this phenomena is called reflection. And due to reflection property of an object, it is visible. The second one is called transmission. Transmissions refer to the passage of light through an object. If two objects are placed side by side and one object transmits light and other is not, then the first object will be darker than the second object. It helps to generate contrast. The third one is called absorption. If a light rays neither pass through nor bounce off an object but are taken up by the object, this phenomenon is called absorption. When all light absorbed, the object becomes black color or dark, and the energy of light will be converted into heat. If some part of the light are absorbed and some are reflected, then the object will be colored. Let's see how. This is the full electromagnetic spectrum, and only this portion is visible to human eye. That's why this is called visible light. The more powerful is violet light and less powerful is red light. If an object absorbed all lights except red, then the object looks red color. And the fourth one is called refraction. Refraction is the bending of light as it passes from one medium to another of different density. The bending of light rays give rise to an angle of refraction. Let's see another example. When light passes through the air, and if you try to see the object from this area, you can see it. But if one glass slide is placed in between you and this object, then what will happen? If you try to see the object from this area, you can't. You have to see it from this area only. Now the question is, why I am talking about refraction? Is it has any importance? You can see another example. A lens is placed between you and object Then let's see what will happen. Two lights are passing through this lens, and due to refraction, the two lights will meet in this area called the focus. If we try to see from this area, a virtual image will create, which is larger than the original size of the object. By using this property, we can magnify an object. Now, the last one is called diffraction. As light passes through a small opening, such as a hole, slit, or space between two objects, the light waves are bent around the opening. This phenomenon is called diffraction, and it will be discussed later. Our purpose is solved now. We can use magnifying glass to enlarge microbes in order to make them visible, and we will further increase the contrast. A greater details, that is the internal structure, can be obtained. Now the question is, if the length of an E. coli bacteria is 2 micron, then how many times you have to magnify it in order to make it visible? The smallest object that an average human can see with naked eye close up to 1/10 of a millimeter, that is 0.1 millimeter. So we have to magnify up to 50 times in order to make E. coli visible only. For further details, we have to enlarge it more. Is there any means by which we can categorize lens according to power? Yes, it is. The property is called magnification or magnification power. The definition of magnification is the apparent linear enlargement of an object when viewed through a lens or system of lenses or in any other instrument. Or simply, if a lens can magnify 1 centimeter object into 4 centimeter, then the magnification will be 4x. Magnification has no unit. It is generally described as x. An ordinary magnifying glass available in the market has 2 to 5 times magnification power. Do you know what was the magnification power of the single lens used in the microscope built by Leeuwenhoek? It is 70 to 250 times depending on the type of lens used. Now the problem is, if the magnification power is 50x or more, then only we can see E. coli. Using one simple lens, it is hard to achieve. But here is the solution. If we place two lenses in this way, the magnification will increase. How? Let's see another example. If the magnification of the first lens is 10x and the second one is 40x, then the final magnification will be 400x, not 50x. Now it is easy for us. Now we can place more than one lenses to achieve that level. But here comes the second problem. If we try to increase the size of an image in your mobile, at some stage it will become hazy. Here comes the second term called resolution. This is the second property of a lens. If we increase the magnification without increasing the resolution, it has no use. It is called empty magnification. Resolution is the capacity of an optical system to distinguish or separate two adjacent objects or points from one another. Is a much more reliable estimate of a lens utility. If I described it clearly, I hope you will understand better. Take one ordinary scale, look at this area. You can see the lines clearly. Why? Because they are 1 millimeter apart. Now, if we draw 10 equally spaced lines within 1 millimeter, I hope no one can able to see those 10 lines. What will happen? All will see a single thick lines instead of separate 10 lines. This is called the resolution power. Or in more bookish way to describe it is, the resolving power or resolution limit, described as small d, is defined as the minimum distance existing between two objects when those objects can still be observed as a separate entities. The unit of resolution limit is the same as the unit of distance. So if d is 1 millimeter, means if two objects are placed 1 millimeter apart, then only the lens can separate them. But if they are placed 0.1 millimeter apart, this lens can't able to separate them. It will look like this. I hope the same question comes in your mind also. Can we increase the resolution of a lens up to infinity? The answer is no. Do you know why? Here comes a name of a famous scientist called Abbe. He described an equation by which we can easily calculate the resolving power of a lens. And from this equation, we will also learn how D is related to some other parameters. So let's check the Abbe equation. Here lambda is the wavelength of light used to illuminate the specimen, and n sin theta is the numerical aperture of the lens. Now what is numerical aperture? Numerical aperture is the arithmetic product of the refractive index of the medium in which the lens of a microscope is situated and the sine of half angle of the cone of light entering an objective lens. It can be described by this equation, where numerical aperture is n sin theta, where n is the refractive index of the medium and theta is the half angle of the cone of light entering an objective. Now come back to the first problem. How you will increase the resolving power of a microscope? The resolving power of a lens depends on two parameters. The first one is numerical aperture, and it is inversely proportional. So therefore, larger the numerical aperture, greater the resolving power. And the second one is called wavelength of light, and it is directly proportional. Therefore, shorter the lambda, the resolving power will be greater. Now let's see how the numerical aperture of a microscope can be increased. Here again there are two ways. By decreasing the working distance, therefore more light will enter inside the lens, or theta will increase, or by applying oil to increase the value of n. And how to decrease the value of lambda? If we use violet light instead of red light, then lambda will decrease. But actually the blue light is used in the microscope because violet light is harmful for the human eye. The wavelength of violet light is 400 nanometer or 0.4 micrometer. So any further decrease of lambda is not possible because that is the lowest range of the visible light. When we use visible light, we can decrease D up to a certain point. So if light is used to observe an object, you have to remember all these points. I hope you enjoy the first part. Next I will discuss about microscope, but part by part. So subscribe my channel and press the bell icon. Share the video if you like it. Finally, thanks for your support.